Dear students, welcome back. This is the official YouTube channel of SRN BGNR Government Degree College, Khammam. I am Dr. B. Sadish Kumar, Assistant Professor in Biochemistry. In last few classes, we have been discussing about introduction of enzymes, second year biochemistry topic. In those previous videos, we have discussed about introduction of biocatalysts and what is the difference between chemical and biocatalysts, nomenclature and classification process. And we have already discussed about the principles of energy, of activation energy and transition state as well. In today's class, we are going to discuss about the last seventh topic, interaction between enzyme and substrate. Before going into details of the interaction between enzyme and its substrate, uh, let us quickly recap the last previous video lesson. The important topic in that lesson we have covered so far is activation energy and the transition state. The basics in this topic are the activation energy means the enzyme actually accelerate the rate of the reaction by lowering the activation energy. Suppose if we have an substrate in this model, we can easily in any chemical reaction, the substrate or the reactant, suppose the energy state of the reactant is here, it must be activated to a state that is known as transition state. So for this it requires some energy input, and that energy is called energy of activation. So once it reaches the transition state, then from the transition state it will quickly converted into products. Okay, the difference between this reactants and the transition state, the energy level of reactant and transition state is known as the activation energy. So from here to here, this is called activation energy. In a non-enzymatic reaction, the activation energy is more, but in an enzymatic reaction, the activation energy is lower. So the because of this less activation energy, the enzyme quickly interacts with the substrate, and it takes that substrate to transition state very easily with minimum amount of energy. And so therefore, because of the reduction in the activation state, the energy required for the substrate to become transition state, it will be reduced. So because of that, the enzymatic reaction is very fast, and it requires very less amount of energy to complete. In today's class, we are going to discuss about how the enzyme interacts with the substrate with its active site. We already discussed that active site is the site where a specific substrate binds to the enzyme. Okay, so that is called an active site. In a enzymatic reaction, that enzyme binds to substrate. Enzyme binds to substrate. So this is enzyme binds to substrate, and it forms an enzyme substrate complex. So here enzyme substrate binding to each other and forming enzyme substrate complex, and then the enzyme catalyzes the enzymatic reaction by which the substrate is converted into products. Once the products are formed, the product will be released from the active site of the enzyme. Now you will get a free enzyme that can be once again utilized for another round of the enzymatic reaction. So this is the general mechanism of enzymatic reaction. Actually, there are three different theories to explain the mechanism behind the formation of enzyme and substrate complex. There are three different mechanisms. Each one is trying to explain how the substrate interacts with its active site on a specific enzyme. Okay, so out of the three theories, the first theory is lock and key model proposed by Fisher. This is also known as Fisher template theory. In a lock and key model, what happens? Every lock will have a specific key. That means a specific key always fits into a specific lock only. One key doesn't fit in another lock. So in that way, a key is always specific to lock and lock is specific to key. That means that key and lock both are pre-shaped. They already have a specific rigid shape. So that is why it is going and fitting into it. The key is fitting into the lock. In the same way, Fisher proposed that in lock and key model, the enzyme will have a predetermined active site. So that means, suppose this is the enzyme. This enzyme is having this active site. In this active site, this substrate only fits because it has the complementary structure, completely complementary structure. The substrate is rigid and therefore the active site on the enzyme is also rigid. So that substrate comes and binds to this active site on the enzyme like a key fits into a lock. So this was proposed by Fisher, and this theory is known as lock and key model of or Fisher's template theory. So according to this lock and key model, the structure or conformation of enzyme is rigid. Rigid means already pre-shaped. That is not dynamic structure. We cannot bend or move the amino acids which are forming the active site. So there is no movement at all in the active site. There is no conformational change of that active site structure. Completely rigid like a metal. Okay, so like a lock. In the if we try to fit the key in the lock, the key doesn't change the structure of the lock. Simply it fits into it. So in the same way, completely rigid. So this is the important point in his theory. The active site is rigid. So therefore, the substrate binds fits into the binding site just as a key fits into proper lock. So there is some drawbacks in explaining this theory. This theory is unable to explain why the activation energy is being reduced in an enzymatic reaction because that substrate is tightly binding to active site. So there is no movement at all. So therefore, the active site cannot induce any catalytic reaction on the bound substrate. So that is the major drawback. It explains only the specificity, not the catalytic activity of that enzyme. So that is why it was rejected. Later on, another scientist named whose name is Koshland, he proposed another theory. That theory named as induced fit theory. The name itself explains the mechanism proposed in this model is that induced fit means the substrate induces the active site of the enzyme. So that means the enzyme the active site of the enzyme is not pre-shaped or not a rigid structure at all, but it is a similar complementary. It is not 100% complementary, but almost similar uh, to fit the substrate. Once the substrate comes into interaction with the active site of the enzyme, the active site of the enzyme will be changed a little bit so that the substrate can fit into the active site. So that means the active site is not a pre-shaped, but the substrate itself is inducing a conformational change in the active site of the enzyme. So that is why it is called induced fit theory. So according to this model, the total enzyme structure is not at all rigid. It is dynamic. The conformation can be changed before the substrate binding and after substrate binding. There are conformational changes. So because of that, the substrate binds specifically to active site of the enzyme, and at the same time, because of the conformational change only, the catalytic activity takes place. The enzyme will catalyze the reaction very easily. So this explained several several things, and uh, it is in fact very well accepted model when compared to the earlier model, that is lock and key model. So here you can see that induced fit model, the active site is not rigid or pre-shaped. It is not rigid and it is not pre-shaped. The interaction of the substrate with the enzyme induces a fit or a conformational change in the enzyme, resulting in the formation of strong substrate binding site. So this is what the induced fit theory explains. So later on, we have one more reliable theory put forth. That theory is known as substrate strain theory. In fact, this theory uh, is also a modification or an improvement of the second theory, that is induced fit theory. In theory, the third theory explains the substrate is strained. The substrate is strained due to induced conformational change. Due to induced conformational change in the enzyme. The induced active site of the enzyme is more complementary to transition state. So this is what the very important point to note down when we look at this third theory, that is substrate strain theory. So the name itself explains that substrate will induce the active site of the enzyme, and at the same time the substrate binding, the active site will induce a strain in the substrate during the binding of the substrate to the active site. So that is the important thing to be considered in the substrate strain theory. Suppose in the substrate strain theory, that enzyme active site is just like the active site of the enzyme which is proposed in the induced fit theory. So that means it is not a pre-shaped or nor a rigid structure. The substrate itself can induce a conformational change in the active site, but the active site is not 100% complementary to the substrate. It is complementary to a transition state. This is the major difference between these two theories. The induced fit theory explained that the substrate is complementary, to the active site. The active site itself is complementary to the substrate. But in the third theory, that is substrate strain theory, the active site is the changing the conformation in such a way it induces a strain in the substrate, so that during the binding, by the time the substrate binds to the substrate binding site, active site, the substrate will be strained a little bit so that the substrate becomes a transition state. So the binding itself is inducing the substrate to become transition state. So that is why the transition state, uh, the substrate is getting that transition state so easily. So therefore the active energy, the energy for activation, the activation energy is very less when compared to the non-enzymatic reactions. This can be easily explained by one particular example. In fact, this is just an illustration. Suppose if we see a reaction A, that is no enzyme. We have a substrate. We have a substrate here, and we have an enzyme. That suppose that enzyme, the function of the enzyme is to break the substrate into two parts. So breaking is the enzymatic reaction in this reaction. So now what happens, without any enzyme being involved in the reaction, this requires a lot of energy to become a transition state. Okay, so that is why this for this non-enzymatic reaction, the activation energy is very high. Okay, but in an enzymatic reaction, the activation energy is very less because the enzyme is reducing the activation energy. Okay, if we look at these two enzymatic reaction mechanisms, these two models, suppose this B is similar to the model that is lock and key model. In this model, the enzyme active site is completely rigid and it binds the substrate like a proper key fits into a lock. So that means the all the sites on the enzyme active site are completely saturated by the sites on the substrate. So we have enzyme substrate complex here, but it cannot produce any products because a stable complex is being formed. But when we look at the third theory, in that is substrate strain theory. So the active site is not at all complementary to the substrate, but it is complementary to the transition state. So that is why once the substrate binds to active site, the active site will induce on the substrate, induce induce the substrate and try to strain it, and because of that effort the substrate becomes a transition state easily. Once the transition state it reaches, it quickly breaks down to form the products. So that is why, because of the induced strain of active site on the substrate, the substrate is easily converted to transition state and then to products. So these three are the theories which try to explain the interaction between the enzyme and its substrate. So let us have a quick review about these three theories. First one is lock and key model. In this one we have to remember only one thing, that is the active site of the enzyme is rigid. And in the second theory, induced fit theory, the active site is not rigid, but the active site is complementary to substrate. And when we come to the third theory, that is substrate strain theory, in that theory it explains that the active site is not rigid, but it is complementary to the transition state instead of the substrate. So therefore the activation energy is reduced. And in the next topic we are going to discuss some of the basic terms usually we may come across in enzymology, and we have already discussed about the general classification of enzymes based on their reaction mechanisms. We have differentiated all the enzymes into 6 classes, but here we are going to look at another kind of classification based on the composition of enzyme. Based on the composition of enzyme, we have two different types of enzymes. Composition means enzymes are made with enzymes are nothing but proteins in most of the cases, except ribosome. All the enzymes are just proteins. Proteins means they will have sequence of amino acid, just amino acid sequence, and that fold upon itself, and lot of interaction takes place between those amino acids, and it forms a globular shape molecule, and that is called an enzyme that specifically catalyze a specific reaction, and it binds a specific substrate. So therefore there are two different kinds of enzymes based on their composition. First one is simple enzymes, composed of composed of only protein part. So that means, suppose this is an enzyme. This is an enzyme. This enzyme is having a complete amino acid sequence only. So when we break the enzyme into constituents, what we will get? Only amino acids, just amino acids and nothing else. So this type of enzyme is called a simple enzyme, because it is completely composed of sequence of amino acids or protein. And the second one is complex enzymes. Complex means they have simple part as well as some other part also is there. So it composed of protein plus relatively small organic molecules or metal ions. So in most of the cases, this enzyme will be like this. This is a protein part which contains the required amino acid sequence. But in most of the cases, this protein structure is not enough to carry out complete enzymatic reaction. It requires some assistance by some organic molecules or some metal ions also. Suppose if we take an example of hexokinase. Hexokinase, the first enzyme of glycolysis. So the function of hexokinase is that binds glucose on one glucose on, suppose this is enzyme, it binds glucose here. Then what happens? It should transfer ATP to a phosphate to the glucose to form the glucose 6 phosphate. And when we discussed about the oxidoreductase enzymes, so the oxidoreductase enzymes, the function of oxidoreductase enzyme is just transferring electrons, either giving or withdrawing electrons from the substrate. So there must be a donor like NAD plus. Suppose NADH is there. This gives electrons to the substrate which bound to the active site. So that means this enzyme is having a simple protein and as well as another organic molecule which provides electrons to the substrate. So this organic molecule in this case is NADH, sometimes FADH2. Like, so these organic molecules plays a major role to perform the complete reaction. Without the presence of this electron carrier, the enzyme alone cannot catalyze the oxidation or reduction reaction. So that is why this organic molecule, now it is called as a coenzyme, the part of the enzyme which assist the simple protein part of the enzyme. So that is why it is called a coenzyme. Okay, so that means it is a complex enzyme. Complex enzyme contains a protein part as well as some organic molecular metal ions. So the functional unit of the enzyme is called holoenzyme. In case of complex proteins, in case of complex proteins, we will have we will call the complex protein as holo, means complete, the complete enzyme. That complete enzyme contains protein part, which is also known as apoenzyme, and a non-protein part also, like NADH or FADH2 and anything else. Okay, so in this case we have that protein part is known as apoenzyme and a non-protein part. If the non-protein part is a organic molecule like NADH or FADH2, what happens? That is called as a coenzyme. In some of the cases, instead of organic molecules, the enzymes use metal ions to assist the catalytic process. So these metal ions like zinc, some metals like calcium, um, potassium, so some metals generally utilized in the catalytic reaction. So these metals are known as, are called as cofactors. If it is a organic molecule, it is coenzyme, or if it is a metal ion, then it is a cofactor. So the next term we have to discuss is a prosthetic group. Prosthetic group means, we already discussed about the coenzyme. So coenzyme binds here and substrate binds here. This coenzyme, if it is bound loosely to the enzyme, it is called coenzyme. Loosely means the interaction between the coenzyme and enzyme is by non-covalent interactions only. Okay, the interaction between coenzyme and enzyme is very loose, very weak interaction. Those are non-covalent interactions. Sometimes the coenzyme may tightly bound to, that means permanently and tightly binds to the enzyme. That kind of coenzyme is called prosthetic group. Prosthetic group is nothing but a coenzyme which is tightly bound to the apoenzyme. So this is called prosthetic group. So we cannot easily remove that prosthetic group from the apoenzyme, but in case of coenzymes we can easily take it out from the apoenzyme. So that is why it is dialysable. The coenzyme is dialysable, whereas the prosthetic group is non-dialysable. So most of the coenzymes are nothing but B vitamin complex group. Okay, we have B complex vitamins, several of them. All the B complex vitamins, most of the B complex vitamins just acts as coenzymes. So that is why they are essential elements. They must be included in our diet. Uh, Suppose, for example, NAD. NAD means nicotinamide adenine dinucleotide. So that means it comes from niacin. Okay, flavin, FAD comes from riboflavin of B complex vitamin. Biotin itself directly involves as a coenzyme in carboxylation reaction. Tetrahydrofolate, that means folic acid. TPP, thiamine pyrophosphate, thiamine, B complex vitamin. CoSH, this is pantothenic acid. So all these coenzymes, most of the coenzymes are simply B complex vitamins. The last topic in this unit is fundamentals of enzyme assay. Usually in medical field and most importantly in diagnostic labs, the enzyme assay is very much important diagnostic tools for the assessment of a patient, whether he is having any um, problem with any functioning of any organs. Like liver function test we can easily assess by assaying the liver enzymes, and in the same way kidney function test, uh, next gastric function test. So all these functional tests which are performed in a diagnostic lab are by assessing some specific enzymes, the activity of some specific enzymes. In laboratory methods, the enzyme activity will be measured on the basis of how much subunit, substrate is consumed and how much product is formed, but not how much enzyme is there. So it will not assay how much enzyme is present in a sample, but how much activity is there for that enzyme. So we will simply measure the amount of substrate converted into product. We measure the activity, not the amount of the enzyme. Okay. So that is the fundamental for the enzyme assay. So in this assay methods, first first thing is we have to use large excess of substrate. So there must not be any limitation for supply of substrate. So there must be very large excess of substrate available. So therefore what happens, if enzyme is more, then the activity will be more, and it converts more substrate to product. So based on the product formed, the concentration of the product which is formed after a particular time, we will find out the activity of the enzyme. If more product is formed, then that means more enzyme activity is there. More enzyme activity is there, that means more enzyme molecules also there. So for that, we will measure the enzyme activity based on the formation of product. If we use less amount of substrate, then what happens, even though there is a lot of enzyme present, but due to the limitation of substrate, the enzyme activity also decreases. We already discussed about the factors affecting enzyme activity. In that we discussed about the substrate will affect, the concentration of substrate will affect the activity of the enzyme. So to nullify the effect of substrate on enzyme activity, we use large excess of substrate. Suppose we have 10 enzyme molecules, and these 10 enzyme molecules interacts with 10 substrates per 1 second. Okay. So what happens, if we use 100 molecules of substrate, then what happens, in 10 seconds these 10 enzyme subunits will consume 100 substrates. So we will get 10 products per second. Suppose we have the same 10 enzyme molecules, and we use only 5 substrate molecules, then what happens, it will consume the 5, and if we continue the reaction for 10 seconds, what happens, only 5 are there. So that means it converts only 5 substrates to products. So now what we will get, 5 molecules per second. Okay. So this way it is misleading. So that is why if we use 10 enzyme molecules, we may use 1000 or 10,000 substrate molecules. So therefore, during the reaction process, there will not be any shortage in the supply of substrates. So if the enzyme molecules are more, then the product will be more. So this is the basis of assay of enzymes. And the last topic is units of enzyme activity. Because we are measuring the product concentration, not the enzyme concentration, we will use two different types of units. The first one is ketol. So ketol is an unit that is proposed by IUB, International Union for Biochemistry. So the same one which appointed the EC Commission for the classification and nomenclature of the enzymes. So this explains the definition of ketol. One ketol is, or the definition of ketol is the conversion of 1 mole substrate per second. That means simply mole per second. So how many moles of substrate is being converted to product in a given time. So that is the measure of ketol. Suppose if the enzyme is converting 5 moles in 1 second, so that means 5 per second, 5 moles per second. If more enzyme is there, then it may convert 10 moles of substrate to product per the same time. So now the product will be more. So that is why the ketol always denotes that how much substrate is converted to product in a given time. So that is simply moles per second. So another unit is also there, which is international unit or SI unit. It is just similar with the ketol, but only thing is in the ketol we used the units moles per second, but in SI units we use the unit micromoles per minute. So instead of moles we are using micromoles, and instead of second we are using minutes. Okay, so therefore we can convert ketol to international unit or international unit to ketol. So that means one international unit is equal to 60 microketols. And apart from these two units, most of the laboratories are still using some conventional old units like King Armstrong units, Samyoji units. So there are several different types of ways to express the activity of enzymes. So when we look at any uh, result of enzymatic assay, we must consider the units of their expression also, because based on the units the results may vary. So units also important to consider. So most of the research laboratories use either ketol or SI units, but the diagnostic labs sometimes may use some old units also. So that is why we have to be very careful when we analyzing the results of enzyme assay. In these video lessons, we have discussed all the fundamentals pertaining to enzymatic enzymology. So we from with this we have finished learning about the fundamentals of enzymes. Thank you.